Good morning, all. So, uh, it's a privilege to present my thought to the all the intellectual peoples, those who are present over here. It is my again privilege. I was sometime dreaming that I will be the part of CCMB, CDFD during my searching the PhD career. So when I try to putting my talk on this stage, it is a really, I think that I am little bit nervous. Please bear me. So I always believe that I will put some impact if my research come into the field. So, turning a science into entrepreneurship, I never think about that, how I can put the things forward. But I got an opportunity to showcase my inner talent in BRIC Institute only, that is Institute of Life Science. So in Institute of Life Science, I started my after MSc, I started as a laboratory technician with under the supervision of Dr. Amul Suryavanshi. He is a clinical proteomics, and he given me a task of the development of biomarker, clinical biomarker, that is nasopharyngeal carcinoma. I worked there around 3 years, and I failed miserably. So that is a bigger challenge and motivation to go ahead with the next level. Why? Because do you know the proteomics and protein related research is a really challenges, especially if you are looking for the clinical related research. It's a very challenges in terms of sample, in terms of biological different entities, in terms of availability of sample and as well as the number of sample. So we are working on proteomics, total proteome, especially in 2D gel electrophoresis, and it's around 24 centimeter gel. So nowadays it is outdated. So technology has been evolved. So the proteases application is huge, but nobody look about the protease inhibitor application. We all only think about the protease inhibitor is only used for the lab purpose to do the western and all these things, right? So beyond that, there is a lot of application where we try to explore that one. So no need to describe all these things, protease and how this work in the protein to break down peptide and amino acid. However, this cutting enzymes, protease, it's a tedious enzyme. Not only it cut the protein, but also it cut itself. So hope you remember the game where in early Nokia mobile, there is a snake bite. If that will bite, that will be little shorter. The enzyme is like protease enzyme is like that. And I failed when I tried to do the experiment. I miserably failed around 70 to 80 times to standardize with proteinase K. Hope you know that how tedious proteinase K enzyme is. If you keep it minus 20 and take it out back, the activity is gone. If you alicot it also, keep it in minus 20, use for one time, again also it will be may work or may not work. If you load the enzyme, if slightly bubble will come and go inside another well, the other enzyme also degraded. So it's a challenging part how to handle all these proteases, and that's why this protease and inhibitor role we try to incorporate into the picture. So we know that the protease inhibitor are the biological or chemical compound. It is more important to stabilize the proteins and all. And due to this protease inhibitor, you can stabilize your protein as well as yield of the protein and improve the functionality of the proteins. So with protease inhibitor, you can get a full length protein. However, without protease inhibitor, you got fragmented proteins. So I'll try to put one video. So our purpose is to reduce the unwanted protease activity during cell lysis. This is a simple cartoon video of the protein, and protease try to attack on that, and the inhibitor is needed to block the activity of the proteases so that the protein will be happy. So it block the activity and it is a safer. With our biopioneer protease inhibitor, we are introducing this protease inhibitor, which is unique in terms of various aspects, where most of the protease inhibitor you need to keep it in 4 degree or -20. So this innovative product has come up with the highly thermal stable protease inhibitor. No need to keep it in 4 degree, -20. Interestingly, you can autoclave also. Can you think about that? Any protease inhibitor you can autoclave and you can do it? No, we have that solution in the market. So this is the evolving technology where this protease inhibitor is working, and this is not only for the R&D but also in the enzyme industry, vaccine industry, diagnostic industry, where the protein related work is more, and people are compromising their work because of the pricing, because of the low enzyme cost. So amylase costing 1 kg is around 200 to 500 rupees. Can you put the protease inhibitor over there? Ross, Thermo, Sigma, any of the bigger brand? No, because pricing is always a factor. So we are. But amylase is. No, no, it is not a protein. It is not a protein. Amylase, I am talking about amylase. Amylase is. No, no, no. Yes, protease inhibitor is not a protein. So these are the one scientist over KIIT. He is working on the liver tissue, and he said that ki, Vijay, we will not shift our protocol. He is very strict and forward. I say, sir, you just try it that. So after a lot of struggle with the 6 month of trial, and he has put that he has given the data of with their protein without protease inhibitor with Biopioneer, that is one of the MNC Thermo. So he has put on the column that how we are good in case of the protease inhibitor in stability of the protein. Of the 5 days of tissue lysis, you keep it at room temperature, still there is a good amount of protein is over there. So protease inhibitor not only for the protein isolation or Western blot or any of the IP, all these things, but it will be used for cosmetic, proteomics research, diagnostic, biopharma industry, agricultural industry, food and antiviral drug development purpose also. So these are the problem we face during evaluating the protease inhibitor cases, where it is most of the protease inhibitor are not stable at room temperature. Most of them are cocktail in nature. It is a cross reactive with the proteins, and pH wise also it is not stable. If you sometime pH buffer will be increased 10 or some ratio, pH 10 or some above, most of the protein will not working. We are generally working on pH 8 or something, 8.8 or so. Sometimes people are working on the acidic type of protein, pH 6 or 5. At that cases also it is a challenges to put the inhibitor over there. So all these things and toxicity, high cost, cold chain transportation, storage, shipment, to address all issues, we have proposed this USP, unique selling proposition of our protease inhibitor, highly thermal stable, autoclavable, high efficacy, even if not interfering with other proteins. So this is the uniqueness of our product. and we are targeting a wide spectrum like serine, cysteine, metallo. Hardly the marketed inhibitor is targeting 7 to 8 proteases, whereas we are targeting more than 16 different proteases with high efficacy. So these are few experimental where we take the protein with proteinase K, the degradation is there with our inhibitor, that is almost all good as the control BSA. Similarly, we check with pepsin, trypsin, chymotrypsin, thrombin, and many more, where we found excellent result in the real sample with all the cell line taken from the equal number of cell line we have taken. We put the inhibitor with the marketed inhibitor with our inhibitor. We found the excellent result in the case of our inhibitor in the after 30 days of the keeping room temperature. You just see that it is GAPDH equal number of cell as per their concentration. We also put our concentration. It is almost good compared to market. So it is not interfering based on fluorescence based quantification. If you wish to direct take the protein and go for the fluorescence based quantification, you can easily quantify the things also. pH wise and temperature wise, it is also good. This is the comparative analysis of the marketed inhibitor, where we are far good in terms of storage condition, in terms of cocktail and component condition, as well as the inhibitory activity and toxicity and water solubility. Beside that, you just see that it's a number of enzyme where the marketed inhibitor they are targeting all are in green color, where the red color they are not targeting. We are good in that cases also. So these are few citation of our product in molecular therapy, nucleic acid, biofactor, MBio. Many people are using our product in ILS, ICGEB, NII, and all this. It's a reversible inhibitor. So basically, they are the people, industry, R&D institute, and some are trial is going on in the. We have a sign agreement with Indian Immunological Limited, Starsol. These are the bigger company where we try to put our product for the high volume capacity. These are few scientists, those who are really helped me to provide the testimonial. As you know that getting and convincing scientists to proper data, it's always a challenging, because they are the intellectual people. They will ask based on the your data and scientific thought. If you are not convincing, they will not give a single signature. That is a bigger challenge, where we getting a single testimonial from such a big group. It's really challenging, and we appreciate that they are supporting us in different various institutes like IITs, Kanpur, IIT Indore, ICGB, ILS, and many more. This protease inhibitor already we are in the market in the academic, CRO, and all this industrial aspect. Our R&D is going on in cosmetic industry, food industry, agricultural, antiviral drug development, and all these things. So this is our primary focus of biopioneer protease inhibitor in the application of the diagnostic to put the, as it is a thermally stable protease inhibitor, we are marketed in the for the welfare of the blood collection system to stabilize the serum protein. As you know that how much means blood has been collected from a rural area and coming to urban area, and it is degraded, and we are not focusing on the cold chain transportation also. Can we give a solution? And we are trying to solve that one, and we have a very good R&D on that. And
so one of the possibilities is to ship it with biologics.
yes.
Yeah, but the question which is there is that is a question whether the
Interference.
antigenicity of that would get modified.
Oh.
That's one point which is
Definitely we will keep it in mind.
It's not peptide and other things. So anyway, it can't initiate that thing. But then as an inhibition complex, that would be actually very different than the things.
We'll keep it in mind. Definitely, sir. It is in R&D. Initially, we have checked with the protein stability and all this. Definitely, sir.
Check with some of the existing things.
So it is in R&D. Initially, we have checked with the no, sir. It's a proprietary. That is the 2016 onwards. I am working on that almost 10 years. And
It's not approved peptide. You are not. It is not. Oh, it's already revealed in the patent document.
so
Whatever classically
we are not at all using the any classical proteogenesis. Yes. Sorry, ma'am. Yes. Yes. Yes, madam. Chemical synthesis. We are not at all using the any not biologicals. It's chemical synthesized. Yes. So major. No, no, madam. No. We have a manufacturing setup of 50-liter manufacturing. We whatever the high throughput technique is needed, like mass spec or something kind of that, then we are outsourcing the thing, because we have a very good connection with the a lot of research institute. It's always challenge to a startup to do all these things, but whatever the minimum requirements, outsourcing the thing, because we have a very good connection with we are around 98% purity. Yeah. Yeah.
Rematch to do NMR or
No, we are doing with the HPLC.
PLC will get, but the real functional groups are only evident with NMR.
NMR, but NMR is always a challenging, but we are doing the functional testing like gel electrophoresis or protease activity assay and all these things. So we are doing it. NMR, but NMR is always a challenging, but little big molecule. Yeah. Like no cocktail, single molecules. Interestingly, we have established that the separation technology can be done. Little big molecule. No. No. Along with single molecule. But I will say, because it is only outside we are doing with the lysis and all these things, don't require that things. If you are putting for the as it is autoclavable medium, if you are putting exogenous proteases for the
So in your lysis process,
using,
so that at the time of lysis, whatever was is
protected over there. Medium, if you are putting exogenous. Apart from that, yes, ma'am. Using not small molecule, bigger one, but not biologic, synthetic. No, nothing. No. Apart from that,
and you're only checking purity by HPLC.
not small one.
Cannot be
Bigger.
sure it's pure because there will be many things that don't have
But not
absorbance that
synthetic.
they still be there.
No,
that's why I said
So why can't you check by mass spec?
we are not detecting in mass spec. We are going for the mass spec based technology detection bigger molecule. It's a more than 1,000 kilodalton. We are going for the mass spec based technology detection bigger molecule.
By HPLC, 98%.
then also you are there.
Everything that doesn't absorb at 16 nanometers.
Or 280 or whatever you're checking, no, your UV vis
So
detector
you
would have been
FTIR.
set somewhere.
FTIR we are doing.
So
The function FTIR we are doing. No, sir. No, ma'am. We are in the process of NMR in ICGV. That is going on, madam. Because many research is going on. Because cocktail is a challenge. Many people are using cocktail. We are introducing for the R&D initially. If we will go for the antiviral drug and all these things, definitely it will be. Many research is going on. Because cocktail. 1D NMR we have done. No, we are going for 2D also. 1D we got the result. We are going for 2D. I don't know so much detail of NMR technology. We are going with ICG with Dr.
actually require 1D for chemical
1D we got
validation. You require 1D.
1D.
And-
So much
and
detail of,
that
we
stable throughout your storage period of the time. Functionally, you have shown that it is there because it is able to protect. But whether the molecule remains as it is or not is something which is.
Yes, definitely. We have a chemist, Dr. Dindal Mandal. He is. Yes. Actually, my supervisor is a organic chemist. He was in NCL Pune, and he has a good experience at the USA Mayo Clinic and all this of around 12 year of experience. Dr. Dindal Mandal. So I was the PhD over there at KIT. The Professor Mandal is guiding on aspect of this. So I was the PhD over there at
However you're testing purity, it needs to be part of your QC,
yes.
right?
No,
So-
madam. Why you no?
What are you doing for your QC then?
Initially, we are going for the R&D protein isolation. So protein isolation purpose, we are just doing the means synthesis, characterization, and go for the functional analysis. Is protease inhibitory activity is there or not? Once that will be done, we will go for the in QC part only. We just do the HPLC analysis. Initially, for that phase, we have to do the or else batch to batch variation will be come. So in this regard, we are because cocktail inhibitor having the lot of cocktail, we don't know many things about the things, but in our point around 98% pure. So we are giving the COA around 98%.
Yeah, actually, my point is still the same.
Yeah.
If you only tell us 98% by HPLC, is that enough? Nowadays, with methods have evolved to the point where if you do a very cheap and very affordable mass spec, will that not be better than doing or
Well,
TRSA for your, as part of your QC?
limitation is that, madam, we need to
A very
go regular basis. It's little difficult. We are usually use the mass of ILS, but sometimes it's challenging to do the mask. HPLC is easily available. Go regular basis.
No, so I think
We are usually use the mass of ILS.
your plan is to put them in clinical therapeutics and other things. So they have a very rigorous system.
Yes, yes.
So please check with them what is the acceptable FDA, EMA, what are their level of chemical validation tools
Yes.
which you must put it in your QC and you may get those machines in your grants.
It's always a challenge to start up to
No,
do
but
the.
if you please,
Yes.
because ultimately any modification of any biologics happen, it may require regulatory approval.
Yes.
Now regulatory approval will look at your data with a very where what both of us are saying is that there simply gel HPLC may not work.
Okay.
And you may be at that moment realize that I should have done.
Will do that. That is going on actually in regular NMR is a little challenging because we will plan for that. We have done our initial study. So 1D NMR we have done, but we'll do. That is going. We are asking about the mass spec. Yeah, we will do that. Challenging because we plan for that. Okay. Have done our initial study. So 1D. Okay. Shelf life is around 2 years. We are asking about the mass spec. We will do that. It's around 100, 10, 20, 21 autoclave activity. Yes.
And it doesn't drop.
No.
Same.
Same activity. It's around 100, 10, 20, 21 autoclave. So these are the concept where we are planning to put over the things. Apart from that,
Shelf life is on
2 year, yes,
on
room temperature. So this is for the protein. Progen, if you keep it in, means your question is, if you, I will keep the 4 degree or minus 20, it will be work or not? Yes, madam, it will work. So this is for the protein stability or inhibitor? Inhibitor. No, madam, it is not affecting. Keep the 4. We checked. No, we clear.
Put it and then they will
No,
work.
it's a basic question where the question, whatever it is coming now, it is very basic. We need to address that one. And we have done since 2016 onwards, we are working on particular this molecule. Okay. So these are R&D is going on for the antiviral shrimp and antiviral bird related things. This is our organization where we are working on that. Particularly, we are very good, well equipped with the central facility at KIIT, TBI. So these are facility where we are working. This is our manufacturing pilot scale. So this is pilot scale manufacturing setup of 50-liter manufacturing where we have synthesized our product. This journey started since 2016 onwards. We are a ideation stage. Ideation stage to prototype stage, it is taking a long time because during that time I was in PhD student. And I started my company in 2020. Then we got some few grant from BIRAC and we have validated our data with field trial support of the Startup Odisha grant and product launch we are around 22 December. And we got the CBRI grant for upscaling and then seed fund for market expansion and all. So in this way, we are around 10-year of journey where we are try to explore more on the particular product and improving day by day. Not only protease inhibitor, we have a list of product, but it's a regular product as you are using. We have a very good collaborator at Gujarat, so we are collaborating for some other product also. Because when I come to a single product, people are asking about what's next. It is a different type of question. We cannot address every question. But thing is that with collaboration, cooperation, we can go ahead many more. I appreciate a lot of effort has been put by our scientists, but to convert that one into the venture is always a difficult where I face. So this journey, I really supported by these peoples. It's not a single man journey. It's a journey of the people. It's a journey of my mentor. It's a journey of my friend. It's a journey of my junior, my all the staff and the mentor, those who are guiding us. So these are few publication highlighted in articles, not publication. So these are we exposure in the global level also. We are some achievement in the winner of the tandem is one of the achievement, winner of the NASCOM is one of the achievement along with the invitation from Mahamahim Rashtrapati ji. So beside that, so these are few photo and I'll little try to put on the how you can do the things on after R&D to a venture. There is a lot of scope on that. So my incubation center is KIT TBI. So I'll just say that. So where we award grant not happened by chance. We are getting a lot of grant, but it's a lot of hard work behind that and lot of grilling with that also. So based on that, we somehow we say that key success, but success is not a fixed point. You can go beyond that also. And funding opportunities are more. Now government is giving the ideation stage, prototype stage, proof of concept stage, testing and commercialization stage. So this is very important for you peoples, those who are in the R&D and try to convert your idea into the commercial venture. So government is now putting a lot of effort on that. So this is very important for. So SIP is one of the program where 10 lakhs is government is giving. There is no need of putting any venture on that. You just give your idea. Based on your idea, they will pick the things and you can go ahead with that idea of 10 lakhs rupees. After that, there is a big grant is there, whatever we are getting, it's around 50 lakhs grant. And you can validate your things of idea into the next level. And you may get the other ministry, like suppose your component is related to electronics, there is a mighty grant. The Ministry of Electronics and IT. Not only BIRAC is one of the agency. Apart from that, there is a lot of agencies are there. So as we are a biotech people, so we think about the BIRAC and all these things. But beyond that also, many ministries are there, those who are supporting. Startup India Seeds funds are there. And many funding opportunities are there. And one of the testing and commercialization scale, we have a CBRI. I got the CBRI also. So this grant also very important. Then venture capitalist also there. It will come next by next. So when you see that now beyond that wall, you may not visible. When you cross that barrier now, you can see the other world also. So my suggestion is that you need to cross the barrier, and crossing the barrier is a real challenges. And even if as a scientist, we only think about publication, after that we may not think about how we can go ahead. So that is also stuck in my mind, and regulatory, all the approvals, how to handle the questions, many things are there. We need to overcome that one, and it's a real challenge. And hopefully if I can, then you can also. We are in the process. We are in the process. We have a 2-3 angel investors. So among them we have discussed. Apart from that, this is the way where your stage will be evaluated. So this is very important. Ideation stage. What is ideation to prototype? Prototype to validation, and validation to deployment. How you can deploy? Whenever I go and ask to the people that this is my product, and you can try with any of the brand, bigger brand. So that deployment is always a challenge. I need to showcase that these are the data. I open the notebook or laptop. I show that these are the data, these are the comparative analysis, these are the things. Then they will try. If they will satisfy, then only you can got order. So that is converting the order. Purchase order is a very challenging. One purchase order, even if you 1 rupees, if you done, then you have achieving 0.0001%. Another next level of round is there. So we need to do the things with the commercialization and all this program, we can go ahead with the things. So what is challenges in bio-entrepreneurship? As you all know, it's a high costing R&D, definitely. As well as intellectual protection, market acceptance, adaptation, talent acquisition. These are the major challenges. Many people are come and join with me after learning many things. They need to shift. It's not like R&D, PhD like things. We are a fix. We are need to get the degree. So we have to do the things. It's not like that. That is a bigger challenge for a startup, especially in the deep tech startup who is working for R&D on biotech segment. What I face also, but fortunately, I got a very good team, dynamic team, and especially the girls, the women's. So the negative part of the always in the mind, अरे लड़की क्या कर लेगी, एक लड़का होता तो अच्छा होता, ये सब it's coming. But I blessed that we have a fortunate team member like Swati. He left after 4.5 years, but he developed many things. He is a dynamic. So he is in other company. Obviously, they need money. They need, and you cannot support in everything. So talent acquisition is very important. Ethical and social concern is very much important as a scientist. If you are not socially concerned, you cannot get a product into the market.
happen when the staff leave?
So, we believe on the people, sir. No, we are not stringent guideline. If you are not socially concerned, but people know that if you are a strong relation with the people, people will believe you and people will be protect their things. Belief system is very important. If you are not believing your student also, it's a challenging. If you are a strong relation, so that is one concern. These are the most of the clinical trial, data privacy, security. Most of them are lot of challenges. The question arises: what is the solution? May I give the solution? These are the most of the clinical trial, data privacy, security. Seriously say that ki, I don't have solution. You need to find your own solution. And how you can find the solution? Build your leadership within you. That is most important. If you build the relationship, motivation, ethics, influence, all these things, characteristics, if you build, then definitely the entrepreneurship journey will be smooth and all these things. So, may people agree or not, honestly saying that you are developing a very good molecule or very good therapeutics things, it is only 30%. Rest 70% is a venture development and all these things. And you need to adopt that one. That is very much important. As a scientist, when we got a single gel picture after 70 time failure, we are excited to make a molecule. It's not like that. So 70% the venture development is really challenging. We need to learn that things. It is not in the textbook, but people are trying to make in the MBA program or some other program to learn all these things. And you should be a broader and mindset of all these things. So finally, I really acknowledge my gratitude to the Host Institute, CDFD, and the Incubation Center and team members, especially Madam Dibya and all the faculty member and of the scientist fraternity. And finally, I will say that success is a journey, not a destination. So thank you. Any question, I'll happy to address.
So I understand that in preserving
Finally, I will say that success is a journey, not a destination.
biologics and other things, there is a significant impact which people can foresee, right? But what was your take on, because I think you mentioned diagnostics also in the
Yes.
thing. So what are your plans on diagnostics?
Diagnostic, we are planning for the initial testing of, initially we have done, especially in the protein degradation, serum sample degradation, stability, interference check, all these
Okay.
things we have.
You are, okay, so that means that you are saying that, my role is in sample preservation,
Preservation,
especially the proteose
protein related.
preservations. That is the sample preparation. So you think that you can add a component and things will become this thing. Okay. Preservation, especially the proteose
Protein
related preservations. That is the sample preparation. So you think that you can add a component and it will become.
Acha.
तो उसका कुछ प्रिवेंशन लॉ है या कुछ है?
Sir, honestly telling, I am ये सब जो क्वेश्चन आता है कि मेरा आइडिया कोई चोरी कर लेगा, 1000 of idea will coming in 1 minute, sir. कोई चोरी नहीं करेगा सर, जब तक आइडिया will not deployed. If the idea will be deployed, then you will be far ahead. अगर far ahead हो गए, आप 2 कदम 4 कदम आगे जग गए ना, जो पीछे आएगा पीछे ही रहेगा. You will be the front runner. So आइडिया वैसा नहीं है चोरी हो जाएगा. होने से क्या होगा? सोसाइटी को तो जा रहा है. Who care about that? I am not caring about that. If somebody is taking my idea, honestly speaking, many people will come and go. And if you are a genuine, if your brain is genuine, then you can make another idea. Failure is just one part of the things. So I believe that if you have an idea, you just share the idea. If you share the idea, some people will guide you. Like the scientist who has guided me, because I share the idea. If I will not share the idea, if I will confine within myself, then I will not get the input from these guys. Guiding, like the scientist who has guided me, because I share the idea. If I will not share the idea, if I will confined within myself, then I will not get the input from these guys. इनकम टैक्स के लिए सर मुझे पता नहीं था, 3 साल के बाद पता चला कि 35% का इनकम टैक्स देना पड़ता है. सो इनकम टैक्स इज वेरी डिफिकल्ट सर. चार्टर्ड अकाउंटेंट बहुत अच्छे से बता देगा. स्पेशली इनकम टैक्स इंडिया का सिस्टम क्या है, देखिए आप एक कंपनी करते हैं तो 20%, 18% जीएसटी दे देते हैं, राइट. यू मे कंसिडर एज ए 20% आफ्टर all the expenses, everything, to give the salary, give the rent, to do the experiment, to consumable equipment, everything. You need again, if you making profit, you need to pay 35% to the government. Give the salary, give the rent, yes, madam, to the experiment, to consumable equipment, everything. You need again, if you making profit. Generally लोग बोलते हैं R&D में सबसे बड़ा चैलेंजिंग है. मुझे तो वो फील नहीं हुआ मैडम. R&D में तो हो गया, क्योंकि R&D में दोनों बच्चों को पूछा, भाई ये कर लेंगे, मैं ऐसा किया हूँ, I got this result, you can try it. If I not validate myself, I validate with 5 people. If their hand is good, then product तो चलेगा भाई. दोनों बच्चों को ऐसा कर लेंगे, मैं ऐसा किया हूँ, I got this result, you can try it. If I not validate my friend is there, is Dr. Rohit Kumar Singh. Actually, he is the inventor of the molecule, but I save the application part. So when this is not my PhD work, this is the part of our correlation PhD work. So when he said that कि भाई ऐसा ऐसा है, सर ने बोला अरे ये काम नहीं करेगा, you can this data will go to dustbin. पेपर पब्लिश नहीं हुआ, 2-3 बार रिजेक्ट हो गया. Rejection is a good part because it is meant for product. When he said that कि भाई ऐसा ऐसा है, सर ने बोला अरे ये काम नहीं करेगा. So other step, especially in terms of we cannot sit in the AC room and we think that my product will be sold. Be honest answer. So you need to travel a lot in flight, in maybe train, in maybe taxi, in maybe single pair में ऐसे ऐसे करके जाना भी पड़ेगा. वो जर्नी हम लोग कर रहे हैं मैडम. Still now we are struggling to make a single product to the marketable. It's not a easy journey. If you are coming with the I am made in Japan, we are made in Germany, people will happy and take the things. They are not asking any question. If I will come from India, best product, then people will ask me the question. And hell number of question, you cannot address that one also. So that is bigger challenging part, and that we accept positively and we try to implement in the positive way. So that is very much important, madam. So especially in the other product segment, market commercialization is maybe easy. Especially in the biotech segment, the market and commercialization and penetration is need a proper commercialized guy. And we are not good in the commercial, but we are putting money on the discount structure with a distributor, partnership, and all these things.
So this is a transition problem between, so most of the people are like, they like somebody should come and buy.
Yes, sir.
You have to actually convert your idea, monetize them, and all these things. Find value for them.
Yes.
Sometimes they are elected, they don't want to get into that. But you can say, if you do it, you will get
Benefited, yes.
And I am there at the back. If you agree, come and work with me.
Work with me, yes.
So I have a question. Actually, I have two. But the first one is, if right now you are trying to market a little bit to academics, and that's a very, very small market. You mentioned that you want to take it to diagnostics, where Akash already pointed out that you've then got to make sure it doesn't interfere with downstream assays. But if you're trying to market this to biologics, right? Stability of biologics. There's a whole thing that it
We
has
are in the trial, madam.
You are going through preclinical trials on
Biologics.
yeah. What are you? How are you not going to do any kind of testing for safety and
Safety already we have done. So
Okay.
whatever the toxicity and all this analysis we have done in the preclinical model, we have done with the 114 days of the mice with the highly concentrated doses.
Mm-hmm.
So we are not getting any things response on the physical parameter as well as the immunohistochemistry based study like liver acute
There's no
toxicity.
toxicity.
Subacute toxicity also we have done. Initially, we need to prove in your own house, in-house first. Then you can go for the GLP because GLP grade, if you are going now, it will cost a lot of money, and we cannot afford that things. Initially, we need to prove in your own house. Yes. IITR, we have discussed with that, and yeah. And we cannot afford good amount, ma'am.
Nothing is free.
Nothing is free, and that is good also. We are appreciate that one. If we are make yeah
See the 80%
good amount.
so that once
Nothing
you
is-
start
And
commercializing or
yes, madam. Already we have commercialized.
Yes.
We are. GST is whenever you
When you are getting the grant initial level, then you have to only manage your own people paying, rent paying, all this, no?
yes, madam.
So how do you manage that?
GST is,
Like, did you keep any chartered account or anybody
Obviously.
accounting?
when sorry, I will just say that when I try to put my company, I can I make a company? First question arise. Because if I will make a company, private limited, the legal document and all these things is a huge. So you need to put a chartered accountant, and chartered accountant is responsible for the ROC filing, GST filing, all these things, even if audit, everything. So you need to appoint a auditor also. So many things are there. But minimum costing of in Indian scenario, the Delhi Valley में तो और ज़्यादा है. Bhubaneswar is tier 2 city, so somehow we got good auditor and all. So we are managed to 20,000, around 20,000 annually. But that is also a barrier. क्योंकि एक साल में 20,000 कैसे दूंगा? कंपनी तो कर दिया, बट नेक्स्ट 20,000 कैसे दूंगा? 18 महीने के बाद तो प्रोजेक्ट खत्म हो जाएगा. How I can give another 20,000? These are the barrier. But whenever you overcome that barrier, na madam, so you will do that कि ना, we can make a big business. You have a clear mindset कि अभी हो जाएगा कुछ ना कुछ तो.
So you need to funding factor.
Funding requirement, sir. So you will do that कि ना, we can. No madam, funding is a problem. How we can say that even if scientist can funding issue, then as a startup, funding required. Private funding, no. Biotech में तो कोई private funding नहीं देता है. Although we are at TRL 9 level. Even if scientists can funding technology readiness level. So our technology is ready up to 9, means we are commercialized the things. We are a pilot scale setup. Although we are at TRL 9. It's depend, madam. If the even if your technology is TRL 3 also, investor is putting money. Like cancer related drug discovery and all these things, because depend, madam. If the even if your technology is TRL3 also,
When
investor
you
is
have
putting money.
this
Like
company
cancer
which is
related drug
accepting your
discovery
product
and all these things.
as a
Because-
part of their system, and they have done already due diligence, right? Some of the people in that company or the things, and they are looking for an opening of engine financing and other things. They already know your effectiveness.
Yes, yes.
So maybe you can pitch these ideas to them to convert them into. So there you have a committed buyer. At the same time, they have done their this thing, and they are more likely to be a thing. This is some of the strategy ideas. Some of the, I mean, at least I know some people who have tried that.
Please, sir, communicate. It's a great use help for me. So
That's one of the.
So that is called strategic partnership, strategic investment. We are looking actually. So we are looking for the people those who are really helpful for the both the angle. Because if you will invest on me, not me, for BioPioneer, I am not a part
Yeah,
anything.
of course.
It's a independent entity. Don't think that if I am a founder, that means everything I'm not nothing. My CA simply say that Biopioneer is a separate entity, you are nothing. You just found it. So this is not attachment, but if somebody is invest on Biopioneer, if they are looking for the business out of that, then it is a very good business.
Sir, what is your capital investment and what is your working capital on a daily basis?
Working capital is around, now my team size is around 5 people, not big, but there is a one PhD guy who is self-motivated. He never want to go postdoc or something kind of that. He just come and say that ki whatever money you will give, I will join with you. Because he attract by myself, means due to me he has attracted. But many few people are there like that ki who can take the ownership. Some people there is a clause, bhai kitna dega, मैं तुझे input दूंगा. So it's a kind of that. So in terms of financial capital, means capex, we are every year we are burning more than 50 lakhs rupees. So in terms and other capitals also there, like few of the technical things like filing patent, putting the things in the, all these things are there, traveling and all. Others are there. Around 50 lakhs is annually we are. Means it is a minimum. If somebody say that ki 50 lakhs, 50
So
lakh
in
kuch
the
hai."
intervening time, when you have just finished one grant and you are awaiting for a next grant, there's a gap. That gap, what do you how do you deal with that gap? Number one. Number two, I mean, I hope there are one way is that I imagine that you could loan to the company, right? Because again, it's a loan because that can get that.
It's always a challenging, sir.
Right.
Is a
I understand that there is a challenge, but loaning is one way. As long as the reason why an idea needs a driver is because when somebody has thought through the process, some whenever the person will face difficulty, he knows that there is enough meat in that to overcome. This is only transitory. The other person who copies the idea has no faith in that idea,
Neither.
so he will leave the boat
board.
the first trouble he sees.
Yes.
So that's why it is difficult to kind of replicate, and it's always good to identify, support people, because this is also a network. And I guess that is something which is there. So I think it's a good idea which you have right now, at least in the protection things. Only thing is that you have to
No, we
partner
were
with the
right people.
right people, and who could take it to a much larger scale, wherever these things. And obviously when you are dealing with clinical materials which is administered, so because even the molecule will not be examined, but their formulation will be examined, right? So though you have follow formulation thing.
Yes.
And also individually you saw that it is not toxic and other things. So that would take care of that. Right? So yes.
In the transition period, that whatever question you have said, that is as a founder, we need to, that leadership skill is very important, where I have a very minimal requirement. I try to push myself very minimal accessories or traveling expenses, everything. We need to
No,
switch.
I'm not asking about that. So there is obviously you
No,
are.
if you are putting that
Right.
small effort,
Right.
you are saving the money.
Yes.
Then that money will use for your employee.
Right.
I am thinking in that direction when we have a transition period of
But
1.5
you never
years.
had a situation where you could loan to your company.
Interestingly, we didn't take any loan.
No.
Although you buy a pioneer,
You didn't take outside loan, but
in bank loan also.
if you are, my question is that your company. Yeah. Over the process, have you loaned your company a money? That's my question.
Personal money we have to.
Oh, that is
No, but that you have to show it as loan. No, I mean that you are not showing it as equity.
more capital. So
Capital is a different thing.
loan.
The loan money you can take back. You can
So-
also take back the interest.
So we have spent around, it's from my pocket, it's more than 20 to 15 lakhs
Yeah.
rupees. So during the 2022, that COVID one, when I finished my BIG grant and
My question is, was it a capital infusion or was it a charge?
capital.
No charge. So it is gone means gone. to the
Gone
bank.
में घुस गया मतलब आएगा नहीं सर
No,
वो
no. I mean, loan का तो provision है कि when you become profitable, you can take it back.
वो तो बात का बात है अभी तक प्रॉफिटेबल लास्ट ईयर हम लोग we have a
Yeah, because you
we
have
have so that
20 lakhs.
this,
Whenever it goes, then you can take it.
take it back. Whenever you are profitable, you can take it back.
वो तो टेंशन हम नहीं लेते हैं हमारा जो चार्टर्ड अकाउंटेंट है उसको दे देते हैं भाई तू जो
No, no, that is a
बट वी
different
नो
thing.
दैट कि
But you see, the reason I am saying is that, see, this is a thing which many of the science people don't understand.
दैट इज
Okay, because they have to appreciate capital infusion from loan infusion. Okay, so
we know that
but the venture holder. Then I am saying that broadly.
Yes.
Right.
Once you,
Okay, because they have to appreciate.
once you make the company, and then you are waiting for your first level of your first stage, you're waiting for the ideation grant or all this.
No, madam. I just put my idea to the
Huh.
Bairak.
Huh.
At that time, I was the final year of PhD.
Okay.
So in Bairak BIG, most of the scientists are there, and I was put the idea, but they are appreciate that we need to support this idea because we have a solid preliminary data. Based on preliminary data,
So that time you did
after
not
I
have.
got the grant, I make the company.
Okay. So once you make the company, you had to, whether you have grant or not grant, you had to pay something like some charge to.
Yes, ma'am.
You
Nothing
had.
is free.
No.
Incubation will take.
No, I'm not saying that. I'm saying that once you make the company, but you are waiting the grant to come,
Huh.
and then you incubate. Then you incubate.
You-
Was there any expenditure?
Oh, we
End of the big grant or company foundation or
starting after I got around January 2020 grant means release money has been come to account, and I formed the company in June 13 2020 in the peak of the COVID.
But now I'm seeing that most of these they are asking the startup. So that you had to form a company, or you need to have a startup means you need a company.
Two things, madam.
Hmm.
If you have a company in hold, that is an advantage. If you are an individual also, there is a scheme like SIP or and other schemes are there, which is government is supporting, like Nidhi Prayas also there. So Nidhi is a very good scheme for the supporting the startup. That is the advantage.
I guess what
If
she
you are
is
an
trying to
individual
ask
also,
is
there is
that,
a
like,
like SIP.
when to actually register a company?
Government is
When you have some
Nidhi Prayas.
business model in place,
Very good scheme.
how would you make money?
My experience is telling that ki after getting grant, I formed the company. But many people are doing, they form the company first, then they go for the grant.
That's the right thing.
Right. My experience is telling that
Can you please
because that is also a problematic. If you are putting your name in the grant or something kind of that, then money will come as an individual. If you are putting the company name as a grant, then company account that will be.
Different.
Yes, sir.
So scientists, they allow them to participate. Whereas as a third party institution not affiliated to scientists, you have to be a company to get the apply.
Apply for that.
So that's what my question is, that when you register a company, but you are writing for the grant. And by that time the grant comes, it becomes 1 year. So that 1 year, do you need to pay something?
Apply
to or it is not? Because you form a company.
No, madam. Means income tax and all these things, na. But only chartered accounting, जो chartered accountant है, वो उसको in capital gain you will no need to pay anything, no need to pay. And GST, whenever you will selling, then only you pay the GST. Again, GST again a balance it will be. Whatever you are consumable procuring, you are paying the money to them and against the GST bill. So GST is in your account. Suppose 50,000, 60,000. input credit is there. If you are selling something and you take the GST from the customer, na, that 18% also in there, so it will be balance kind of things. Whenever it will be completely, suppose 50,000, it will overshoot that one, then you need to pay the things to the government. You take the GST from the customer. That knowledge I don't have at that time, so I lost a lot of GST input. Suppose 50,000, it will overshoot that one. Pay the
This accounting, how to do
I myself manage, because that is a bigger challenge. You cannot put for the things for the you cannot put a accountant for that. It's a simple. You just take the invoice and all these things, keep managing properly, put a Excel. It may take 5 to 10 minutes, but it is a regular basis. You have to practice that one. And again, the you try to put that, always pay the money in the bank account only. Keep managing properly. It's a very transparent and clear cut, so that you can have a accountability. And accountability is always be a good part for the any leader to go ahead with the entrepreneur. Check the books of account.
Dr. Bijay, just for the benefit of aspiring entrepreneurs, what sort of agreements should be in place at the time of floating the company, like founders, co-founders agreement, or any sort of agreements?
It's depend, madam, how input they are giving. If somebody is founder and co-founder, if somebody has the idea, but they are not actively participated in the real world, then the shareholding is different. If somebody, if you think my idea, I'm a scientist, and somebody will be commercialized, because they are added value. Without commercial angle, you cannot succeed. So you put little bit more value, means shareholding to them. Maybe as a scientist perspective, I'm telling, not as a businessman perspective, because I am from that background. Then the idea, because if you are a multi-crores company and your value is 1%, that is fine. If you are a 100% means shareholding, but you are not getting means any rupees, that is a valueless. So we need to think about how you can flexible on that.
So you have drawn up agreements before it.
We have a drawn up agreement. We have many people, those who are in the advisory role. We are give a letter to them for the things, and if you, mutually we are agreed to, if they will create the value and money in that, then we will share something. So it's again a transparency mode. We need to pay the 10% tax of TDS with account only. The value and money that we will share something. So it's again a transparency mode. We need to pay the 10% tax of TDS with account. For the goodwill only, they are giving the things. We are not taking lot of things. No payment, because most of the scientists are very good. They are try to help, because they know that how struggling science is, and how we can motivate the people, those who are taking the science to the next level. So that's why many scientists are helping me. I'm not blaming anyone. Always they are helping. We are doing the crystallography and all these things with the collaboration of ICG. So to do more mechanistic study, mechanism, we need to find out how it is binding, where it is binding, what is the concept. Because it's a R&D is a long-term process. It's not a short-term, but simultaneously business also important. Study mechanism, we need to find out how it is binding, where it is binding, what is the concept. Because it's a R&D is a long-term process, it's not a short-term, but simultaneously business also is.
We have to protect our institute because you are giving your time, you are giving institutional resources. So with all the collaborators you have, like ICT or work with PIT, etc., have that institutions played any role in signing any agreements with you, or how has that been?
In TBI incubation center, we have an agreement to access all the equipment and all these things. but whatever the consumable and all these things we need to pay. But in terms of the collaboration, we just try to put that the collaborative work, how in Indian scenario it is going on for the publication, patent right and all these things. And commercial right, we will keep it with us. But patent right, if you are putting the lot of effort in CDFD, then CDFD will be the patent right, or some part of the ownership will be the CDFD and some of the part of the ownership will be the company. Because company will
Obviously, the Indian ecosystem and I am aware of some US ecosystem also. Right from the day one, entrepreneur does not need to pay anything from the pocket. There are investors floating around trying to find a big idea, and they will pay your salary also.
Yes.
Investor. This is not there in India.
In India.
In India, you have to struggle. You have to really convince people, generate a market. Then the established business people will run around to get, I want to buy your business.
Again, that is again there is a exclusive right, non-exclusive right that will come into the picture.
The thing is that how to assist people who are assisting the scientists. See, this inhibitor is a valuable community to any scientist, okay, for the research purpose.
That will come into the
Obviously, you could work out some barter deals with them that, do this work, we will supply you X amount of
free trial and all this.
I mean, we'll supply X amount of things which will work out both win-win, because you will not charge. You will say it as an expense, but not charge it as GST per your book.
Nothing.
And it will take care of that. So that is the kind of deal which you can work out.
We are working on that only.
That kind of thing. So that
even if we are providing through the distributor also.
So the challenge is this. Right now, you have to become, what is it called?
We are working on that.
Called that
Junction.
Where you make some money, right? To show it in the thing. So you have to reach that point, and then you will break even.
Break even.
Right. So that is the thing. So
Yeah.
that's a challenge for Indian entrepreneur, I guess, because we don't have the same ecosystem.
No, we have a very good ecosystem.
What I'm saying
In US and India.
The reason why Singapore they go and the US they go, because bulk of this is struggle part, because there are supply investors are good at the
Startup.
startup. So they are really hanging on and trying to, if you give them a good enough justification, they are willing to support.
Yes.
With the money. And also there is, I just said, I have seen in a YouTube one of the video for people who are interested in. There's a way of pie model of how to share your startup thing. There's a way. So there they say that technical skill is only 2 times. What is the promise? 2x will be the share. But if somebody funds you early, that is 4x.
Yes.
Because anybody paying money is more important than the who is contributing.
The technology, technically, yes, you are right. So that's why the valuation of the company is initially very low. So you may consider as a 1 CR or less than that also people are try to dilute, because if that will come, even if 25% dilution, you can get a more than 20 time, 30 time. So that is a bigger, so high risk, high gain over here.
So they advise, if you are a long-term entrepreneur, start at 2 companies. One is holding company, which holds the patent right. And the other company is the company which you are
running.
And holding company is ANSI. So you can spawn multiple company under the holding company.
I request any online participants if they have any questions to the speaker. You may please unmute and ask. Request any online participants if they have any questions to the speaker. You may please unmute and ask.
Okay, so if there are no more questions, we can conclude today's session. That was really inspiring and insightful. A huge thank you to Dr. Vijayan and Panigrahi for sharing his ideas and thoughts and telling us how science and entrepreneurship can go hand in hand to shape the future. Now, I request Dr. Rachna Bhandari, staff scientist, CDFD, to kindly hand over a token of appreciation to today's guest. And entrepreneurship can go hand in hand to shape the future. Now, I request Dr. Rachna Bhandari, staff scientist.
Thank
CDFD.
you. Very
Thank
nice
you.
talk.
Thank you.
Very frank. We really appreciated that.
Thank you.
Guest.
Thanks.
Thank you.
Thanks for
Thank you
coming.
once again, everyone, for joining us.
Definitely, madam.
For outsiders who have joined us today, please head to the sixth floor. We will give you a tour of our incubator facility. Thank you. Thank you once again. Thank you to the online participants as well for joining us. For outsiders who have joined us today, please head to the sixth floor. We will give you a tour of our incubator facility. Thank you. Thank you to the online participants as well for joining us.
So can we take online tour also of your incubation facilities? Can we take online tour also of your incubation facilities?